Lisa? Sure, sorry. Yeah, two left. Richer than Abi. That was a nice intercept. Okay, good job. Alright, so now let's do a little bit of recap on mitosis and meiosis. I will ask you questions and I'll invite some of you to come up with something else. Alright? We'll start off with mitosis first. Yeah, so this is the question I posed to you. Let's not bother about microtubules or spindle fibers today. We just focus on chromosomes. Okay? Alright, so first question for all of you, we discuss with your friends. What would it look like on the whiteboard? So I ask you to indicate the position of all these chromosomes during prophase of mitosis. Mitosis, prophase. What would it look like? Okay, you may discuss with your friends, and I'll invite one of you to come up to arrange this to your liking. What would the chromosomes look like? How will it be arranged in prophase of mitosis? Okay? Happy? Alright, what will it look like in prophase of mitosis? Anika, I invite you to go and do what you want to do. What will Anika do? Right? So don't do anything? Dana? What? First time I asked her to come up and do nothing. You're right? Okay, Anika is right. Nothing needs to be done. Alright? In prophase or mitosis, the chromosomes are just jumbled up. Alright? There's no particular arrangement. Okay? So maybe that's first thing we think of. Prophase or mitosis. No particular arrangement. Alright, next. What would metaphase look like? Okay, metaphase may be a bit hard to draw this whiteboard. It's called the shape of the whiteboard. But let's try. Nico, you want to try? Do you need to reach it? Is there another way to show metaphase? Is there a way to show metaphase without reaching too high up? Okay, go for it. Right. Hey, okay, not bad. Maybe I extend out the cell a bit. Now give some space. Yeah, good job. Okay, nice. Okay, Nico is done. I have a question. Why don't we arrange the chromosomes like that? Okay, cannot separate, right? Okay, so that's the logic of it. Why we arrange them this way is so that the two chromosomes and sides can come to two lengths apart. We're using the microtubules. Okay? Alright, so how are they arranged? Mitosis, metaphase. They are arranged in a, we just say singly. Okay, they are arranged singly. Okay, what about anaphase? For anaphase, I'll do it for you. Anaphase, you just pull apart. It's something, I know, your mind right now is like, okay, this doesn't look quite right. Okay? What would be more accurate for interface? Okay, what's the difference, right, between me doing this, okay, you keep it like that, versus and this whole thing like that? Is there a difference? Okay, is there a difference? Okay, not pictorially, but just from the mechanism wise, is there a difference is if I flip it this way versus I turn it and then I pull it. No difference, right? Okay. So which one actually happens? Okay, more of the flip one. Okay, but the reason why I'm not doing the flip here because look very ugly, become black suddenly. Right? I want it to be red and blue, I want the vibrant colors on the bottom. Okay? So we just leave it like that for today. Okay, so after interface, so what's the main thing that happens in interface? Sister chromatids separate. What happens during telophase? Okay, during telophase, the nuclear envelope reform. And then there's cytokinesis, and then you get 2 cells. Okay, now we're going to compare with meiosis. I'm not, notice I didn't put in a lot of details like what's in the textbook. I'm only dealing with the differences. Okay, the key ones that will come in useful for your essay. Anyone can recall the different phases of meiosis? Okay, idea prophase 1, metaphase 1, anaphase 1, telophase 1, and go through around cytokinesis. T2, and then another round of cytokinesis. Okay, so 2 rounds of nuclear division. Let's compare and contrast now. Okay, let's start from the top. Let's say we have the same cell again. I have the chromosome here. I have this chromosome here. Okay, so the question I'm going to ask you now is this. What would prophase 1 look like for meiosis? Okay, you can discuss with your friends before coming up on the whiteboard. Okay, what would prophase 1 look like for meiosis? More importantly, how is it different from prophase or mitosis? Ready? Alright, what does prophase 1 of meiosis look like? Which one? Would you come up to rearrange it to your liking? Thank you, Richard. I think our textbook is described as a metaphase state. Alright. Thank you, Richard. Now, do I, you want to do anything with it? Then leave it like that. Sure. Why did you do this? Or maybe you want to describe what you have done. Do you describe describe what you've done on the whiteboard. What's the process? Okay, you join two identical chromosomes. What is it called now? Okay, so actually I think this phase of meiosis, prophase 1, is the toughest phase to grasp. Okay, so if there's a phase you want to pay special attention to, it's prophase 1. Once you overcome that hurdle, it's okay already. Alright, so prophase 1. There are several terms, new terms they will be using. I'm going to write them all on the whiteboard. Let's learn how to use them. First one is synapses. Okay. Next one is bivalent. Next one is chiasma, which is singular. Chiasmata, plural. Okay. And then there's crossing over. And then there's the word non-sister chromatic. How might one use 1, 2, 3, 4, 5 of these words in a sentence? Okay. Have you used it before? Actually, if you tried a quiz and you've used these terms before, okay, how would we use these terms in a sentence? You can discuss with your friends before I invite one of you to verbally just share out how you would describe Wishvan's diagram using these 5 terms. For a start, if you don't know how to use these terms yet, you read your textbook. I think it's important to see if you understand the meaning of it. Okay, then you don't have to memorize. I just realized maybe I'll include one more word, homologous chromosome. What is this? Right. Very loaded, 6 loaded terms. Okay, give you a minute more before I invite some of you to share what you think. Alright, let's begin. I do not need you to complete the entire thing. I'll just invite one of you and then we'll finish the story together. Okay, maybe I'll start off with Aida. You just start us off with whatever words you want to use on this list. When they pair up, synapses, okay, you use one word. So, the pairing up process is synapses. Synapses is a process. You can think of it even as a verb. So one could say, here, synapses occurs. Okay? Hey Arya, you want to synapses with Anika to the canteen? Yeah, so synapses is just pairing up. Okay? I want you all to, so next time if I say, oh, everyone synapses now, and then we'll play Kahoot. That means I want you to just pair up now. Okay, so synapses is just pairing up. So you can just say, here, I have homologous chromosomes undergoing synapses. Okay, Aida, you can pass on to the next person. You use whatever words they want to use there. Raksha. Okay, Raksha, you're next. Okay, when synapsis occurs, the homologous chromosomes pair up, forming a bivalent. Right. Here I have homologous chromosomes pairing up. What is the difference if I pair up this way and I pair up this way? They're both pairing up. Okay, actually, Fishman's method, what he's done here is that he's exposing the sister chromatids of this chromosome to this one over here. This one over here, and this one over here. Usually when we depict pairing up, we depict it this way. Okay? The crossing over tends to be along the entire stretch of one sister chromatid to another entire stretch of sister chromatid. Okay? So normally we will see it this way. I'm actually not sure if this can happen. I don't see why it can't happen either. Alright, so this is an interesting model. Maybe you and I together can go look it up. I'm not sure if this actually happens or whether it can happen. I don't see why it can't also. Okay? But typically what you see in your textbooks, you tend to see the models pairing up this way. Oh yeah. And that's difference number one. In prophase 1, homologous chromosomes pair up. We did not see any pairing up in prophase of meiosis. That's one key difference. We are still at prophase. There's a lot to do in prophase 1 of meiosis. Okay, homologous chromosomes pair up. We are left with 1, 2, 3 more words. How do we use the remaining words? Raksha, you pass on the next person. Yes. Yes, the stop happens. Yeah, hey, sorry, Raksha, you pass on next person. Who? Oh, Junying, okay. Junying. Yeah, look at Raksha with kindness. Right, she's given you an opportunity. How you would like to use remaining terms? Okay, so Raksha said, during synapsis, homologous chromosomes pair up to form bivalents. How would you like to use any of the three terms there? Chiasmata form? Okay, chiasmata form to allow for crossing over. Okay, let's look at that. Okay, so what is, maybe let's talk about what's bivalent. Bivalent is when homologous chromosomes, when you see two homologous chromosomes together, that's bivalent. Okay, so right now if I ask all of you to pair up, your new groups of two, you are bivalent. Okay? Yeah? So this entire thing is a bivalent. Okay, crossing over, what would that look like? Or rather, what's a chiasma? Chiasmata. A chiasma, you watch now, I've just made one chiasma. What's a chiasma? It is that point where it overlaps. Now I'm going to make a second chiasma. Watch, I'm going to make a second chiasma. Second chiasma. Now I have two overlaps. The plural of chiasma is chiasmata. Okay? So there are two chiasmata. If I were to separate this, just one chiasma. Okay, so that's how I use the term. Chiasma is just the point where we meet. But where it meets, it gets quite epic. Okay, so now I'm going to include inside alphabets right now. Sometimes you may see alphabets on the chromosome. Maybe A and a B. Now, question for you. Why is it both sister chromatids have the same alphabet? Sorry? Yeah, they just went through DNA replication, so they should be genetically the same. Okay? Maybe the next question you have in mind is, what in the world do these alphabets represent? Okay? So what do alphabets represent? Now I'm going to untangle the DNA. When I untangle DNA, you'll find that actually they are made up of I need to think fast. T, A, C, G, A, A, T, C. Okay? They are just made up of nucleotides. Guess what? These nucleotides, like words in a recipe book, they prescribe a recipe. Right? Your DNA is a recipe book. You tell yourself what kind of proteins can be produced. So, a segment of this DNA may code for protein. Code for a protein. When I say code for, it kind of means it's a recipe book. This sequence of DNA is a recipe book. It's a recipe to produce this protein. What kind of proteins can I produce? I could produce enzymes. I could produce structural proteins. I could produce hormones. So you realize lots of things can be produced, even though I just said protein. So it goes for protein. It could even call for a protein that determines eye color. Right? So a sequence of DNA that can code for protein, we call it a gene. We have many genes. We have a gene for eye color, we have a gene for hair color, we have a gene that produces enzymes, we have a gene that determines the hair on my skin, we have a gene for everything. But notice this, my eye color is slightly brown if I look in the mirror, the iris. If you look at your own, maybe yours is black. We may all have the same gene because we are all a human species. But our genes may be slightly different. What do I mean by that? So, if small a represents the gene for blue eye color, big A may represent another form of the same gene that perhaps codes for black eye color instead. So, a gene codes for a characteristic. A different form of the same gene, we call it allele. And this codes for the different forms of the characteristic. So what is the characteristic here? The characteristic is eye color. But I can have different forms of the same gene coding for different eye colors. Black eye color, blue eye color. I can have another gene over here, B, representing hair color. But I can have different forms of the same gene. Big B could represent black hair color. Small b could represent brown hair color. We often use a single alphabet to represent a single gene. But I use big caps or small caps to change the allele. Okay? So alleles is big or small caps. A gene is represented by a single alphabet. Yeah? So these are it's the same gene. This is different alleles. Just to test to see if you understand how all these terms come together, I'm going to write some alphabets on the board on this chromosome. Here I have A, looking at this diagram, you can discuss your friends. How many types of genes do you see? Okay. How many genes do you see? 4? Okay, so there are 4 genes. Right? 4 alphabets only 4 kinds of alphabets. There's a gene for eye color, hair color, maybe skin color, maybe height. For this one over here, for the gene of hair color, A, how many types of alleles do you see? 2 alleles. For the gene of eye color, how many types of alleles do you see? 1 type of allele. How many types of alleles do you see for this one? Okay, 1, I saw small caps. How many types of alleles do you see here? 2. 2 alleles of the same gene. Right? So alleles are just different forms of the same gene. Right? If all genes were exactly the same, we would all look exactly the same. But it's because we have different combinations of alleles that we all look different. Because I may have 2 copies of big B. That may result in me having black hair. Another person may have big B and small b. That may result in brown hair. Another person may have 2 copies of small b. Maybe that will result in gold hair. Right? So it's the different combinations of alleles that result in different things you see on the surface. But that's one whole chapter on its own. I hope you appreciate what allele gene is. How much time we have left? Okay, so you pause a while to take out some notes and then digest this a little. Okay, even better, if you like to, you can spend some time talking to your friends about what you just heard. If there's any questions you like, then you bring up immediately. You don't let things snowball here. If you think you understand what has just happened, I have a question for you. Do are homologous chromosomes exactly the same? Okay? Are homologous chromosomes exactly the same? But 2, can they be? Okay? Are homologous chromosomes exactly the same? Ready? Yeah. So, are homologous chromosomes exactly the same? They're not. At least on this whiteboard. You look at this whiteboard, you look at these 2 chromosomes. They are the same in that they have the same genes. but they have different alleles, right? So are they exactly the same? They're not exactly the same. They are the same in that, yes, they have one for eye color and they have one for hair color, but the color different. Okay? So homologous chromosomes are not exactly the same. But can they be? Okay, maybe. What if I had a pair of homologous chromosomes that every single gene exactly the same? Same allele. And it's exactly the same. Okay? So homologous chromosomes aren't exactly the same. They could be, but they are almost never the case. You know why? Along this sister chromatic, there are hundreds and thousands of genes. The permutation right just won't allow the stars to align to have hundreds and thousands of genes being exactly the same. Just impossible. If it were possible, you will find more people looking alike than different. Right? But we all look different. Okay? Why I talk so much about this? Just because we can appreciate crossing over a little bit more. Okay? Just so that we can appreciate crossing over a little bit more. Okay, if you think you're comfortable, then you look up, I will just demonstrate crossing over for you. Okay? You need to appreciate all what just happened to appreciate crossing over and its value of it. Okay, here, notice that these two homologous chromosomes are not exactly the same. Watch what happens when I take this sister chromatic and I overlap here to form a chiasma. Now here's the interesting thing. When things overlap in chromosomes, sometimes they will break off and then they will just exchange parts. It is as weird and as good as Anika, you and I like that, right? You cross the arms over here, right? And then, right, I chop off arm, I chop off my arm, and then we just swap 4 arms. Yeah? That's what happening with the DNA. They chop, and then you know what can happen? They can swap. They can swap the alleles. So, chop, right, chop off, and then I re-stitch back. This can happen. Okay? And this most always happens. And we call that process crossing over. So, if there's another thing you can compare, during prophase 1, crossing over occurs. Whereas in prophase, they don't even touch. There's nothing to cross. Next, crossing over takes place between non-sister chromatids. What does that mean? Okay, these two fellas over here attached to each other, they are sisters. Okay, sisters. However, if I look at these two, they are non-sisters. They are non-sister. Non-sisters, but if they are attached to each other, they are sisters. So crossing over occurs between non-sisters. Now let you do a thought experiment. What would crossing over look like between sisters? It won't change anything, but what would it look like if the chromosome cross over between its sister chromatids? If this is one sister chromatid and this is another sister chromatid, what would the dance move be? Yeah, crossing over between sister chromatids will end up looking like this, right? Right now, right? You cross over yourself. Okay, but that's not what's happening. There is, we cross over between non-sister chromatids. So it's me plus another me, and then we link arms, and then we cut our arms off, and then we swap arms. Yeah, that's what's happening on chromosome level. Alright, so I hope these terms make a little bit more sense now. Let's string it together. In prophase 1, synapsis occurs, which is simply the process of pairing up homologous chromosomes to form groups of 2, which are called bivalents. Where there are chiasmata formed, crossing over will take place between non-sister chromatids. And the alleles may exchange. Okay? So these are how the terms come together. To me, when I was a student, I found prophase 1 the toughest part to understand. The other parts are easier, right? So take some time to digest this part. Okay, in the last 5 minutes, let's look at what happens next. Next, in metaphase 1. Next key difference, metaphase 1. Homologous chromosomes line up in pairs. But in metaphase, they line up singly. And you know the implications of this is if they line up in pairs, that means if I were to pull them apart, I'm pulling apart homologous chromosomes, not sister chromatids. So in this case, we pull apart sister chromatids. In anaphase 1, homologous chromosomes separate. Okay, you see the richness of a compare and contrast question for mitosis, meiosis. You can effectively write, understand the entire topic by writing this essay question. Earlier on in the MCQ, in the output, there was a statement called independent assortment of chromosomes. And it says that it takes place in metaphase 1. Independent assortment chromosomes. Okay, what does independent assortment of chromosomes mean? It means this. Notice that the homologous chromosomes are not exactly the same. So who's to say that this chromosome over here cannot be on the left-hand side instead? Why can't this chromosome be on the left-hand side instead? Whether they are on the left or right-hand side has great implications because it will mean whether it goes to this cell or it goes to this cell. So imagine now in a human being, we have 23 pairs of chromosomes. I don't know what's the math behind it, but 23 pairs, how many probabilities of arrangement are there? Okay, so that's independent assortment of chromosomes. Independently, each chromosome pair can be arranged whatever I want it to be. And that will impact how it goes into each cell. Okay? Alright. Meiosis 2, I have to say blatantly up front, it is just like mitosis. Okay? Meiosis 2 is just like mitosis. The chromosomes will then line up in the middle. Sister chromatids will pull apart. So, if I have to say meiosis 2 is kind of like mitosis. Except with half number of chromosomes. Yes?
Is it possible that there is no crossing over during the M1?
Is there a possibility there's no crossing over during, crossing over occurs during P1?
Sorry.
There's a chance. Yeah, there's a chance. Sometimes very funny, they cross over and then they cross over again. Then like nothing happens. It can happen that way.
Is it rare or is it quite common?
Two? Crossing over?
Not crossing over. Is it
Not crossing over, I would say it's more rare than crossing over. Alright? Hopefully today's lesson was recorded. any point that you felt yourself, your body left, your spirit left your body and it went somewhere else, right? You can revisit it.